Hello students, it looks like we have to resume our online videos for the remaining portion of the syllabus. Earlier our Honorable Vice Chancellor used to inform that as per the mandate issued by the Ministry of Home Affairs in the earlier days, there shall be at least minimum 2 to 3 weeks of classes before we conduct any examination. But the latest mandate from the Ministry of Home Affairs is that there won't be any school or colleges till next 30th of this month. However, the Supreme Court also given a direction stating that we should have completed all our examinations before the end of this month, or at least we should have initiated the examination process before the end of this month. So we are in a very tight situation in that for sure we have to conduct the examinations, but we cannot have regular classes. So in order to complete the remaining portions, I think we will resume our online videos. So before we get into the current videos, we will just recap what all the things we have discussed in our earlier videos. Because in case of the syllabus, we have to discuss about the process of recombinant DNA technology in prokaryotes. Another one is going to be in the eukaryotes. With respect to the prokaryotes, we have discussed for sure most of the vectors and the technology associated with gram-negative bacteria. Example is E. coli. In E. coli, we have discussed about the restriction enzymes and other enzymes involved in the recombinant DNA technology, as well as the vectors principally used is going to be the plasmid. So in order to overcome the limitation of the plasmid, we discussed about the phage vectors. So while discussing the phage vector, we discussed about the lytic cycle. Another one is going to be the lysogeny cycle, in which we also discussed about the in vitro packaging. So from the in vitro packaging, we have understood and created the cosmid vectors. So now these are all related to the prokaryotic vector. The next thing is that if you want to use the recombinant DNA technology and its application in industries, so we have to take all our host along with the vectors into an industrial fermenters. So for an industrial fermenters, E. coli is not the right host. Then we have to use appropriate several gram-positive bacteria. Now again we have to overcome that. We have to create a plasmid which is useful in gram-negative bacteria but can also function in gram-positive bacteria. We call those things as shuttle vectors. So in order to express several important the proteins, so we also need to create an expression vectors. All those things I think we have discussed in detail with respect to the prokaryotes. So in case of a prokaryotes, the major one for the identification of a clone, we used to use a marker, and the marker in all these cases happens to be an antibiotic resistant gene. So we know the significance of resistant gene, antibiotic resistant gene, and also we have used chromogenic substrate, that is going to be the LAC-Z DNA with XGAL and IPTG we also discussed in detail. That is for the clonal identification. So far it is very clear and straightforward. Now we also have to understand how this recombinant DNA technology can also be extrapolated to the eukaryotes. So between prokaryotes and eukaryote, especially in case of a prokaryote, we do have certain advantages. Basically, several eukaryotic genes are cloned in prokaryote host system. Okay, so several genes from the eukaryotes are cloned into prokaryotes, and we have also expressed them in fact. But the regulation of prokaryotic system is totally different to that of an eukaryotic system. So some key genes of eukaryotes has to be manipulated in prokaryotes, but in order to have an optimal activity, we have to clone them back to the eukaryotic system. Now that is the biggest challenge we have it in generating the vectors for eukaryotic system. So in case of an eukaryotic system, we will study systematically first the vectors for yeast system, followed by the vectors for a plant, another one for an animal. So now we have extensively studied in yeast principally because yeast is a single cell organism. It is easy to culture both in liquid medium as well as in the solid medium. That is number one. The another important one is going to be that the cellular biochemistry and regulation of yeast cell is very much similar to that of a plant cell or an animal cell. So the idea is that prokaryotic system is very important because we can clone any of the genes either from a yeast, plant or an animal in a prokaryotic system. We can do all type of manipulations, deletion, addition, create a new site, insert a new site. All those things is possible in a prokaryotes. We can also understand a little bit of a genetics, but in order to optimize the condition for all other regulatory mechanism, it is better to put all these manipulated DNA segments into a eukaryotic host. Now that is the challenge. Now before we get into that, first we will study about the yeast cell and also the genetics of an E. coli as well as yeast. So now today we discuss about the genetics of yeast or Saccharomyces. So now I have given you the pictorial representation of a typical E. coli. Another one is going to be the yeast cell. We focus the major differences with respect to the genetic content. So in case of the genetic material in E. coli, we have a circular DNA, which is the genomic part of it. Another one is we also have extra chromosomal DNA. We call this as a plasmid. This has been identified in most of the prokaryotic cell. Alright. So the genomic material is again a circular plasmid. Another one is that both of them are all suspended in the cytoplasm. Okay. We don't have a nuclear membrane covering this genomic DNA. On the other hand, yeast cell is totally different. So here we only have the cell membrane, whereas in case of an yeast, apart from the cell membrane, we also have a cell wall. Now that forms an additional obstacle during the transformation, but we will discuss that slowly one by one. Apart from that, so this has got a clearly distinguished nucleus with a nuclear membrane, followed by mitochondria. Then the major organelle we are going to find in case of an yeast is going to be a vacuole. Okay. So now this is the mitochondria. When it comes to the genetic component, so the genetic material, the size is approximately 3500 kilobases in case of an yeast. Whereas in case of a yeast cell, it is 4 times bigger. So we have around 14,000 kilobases. Now this genetic content is 4 times larger than prokaryotic or especially E. coli. Now in case of yeast cell, the genetic material, 90% of the genetic materials are genomic in nature. Now in case of an E. coli, it is going to be a circular DNA, whereas in case of an yeast, the 90% genomic DNA is present as 16 metacentric chromosome. So the metacentric related to the position of the centromere present, or in other words we can say that we have the genomic material is present in the form of a chromosome. So apart from that, the other genetic material is present in the mitochondria. We call this as a mitochondrial DNA. And the most important one within the nucleus is the presence of a 2 micron plasmid. It is also called as a 2 micron circle. It is a circular plasmid like that of a plasmid we will find in the E. coli. So such 2 micron plasmid has been identified and characterized in several yeast variety. Alright. But when it comes to the function of a 2 micron plasmid, here this offers an additional phenotypic character to a microorganism, and also it gives a beneficial component to a host system. For example, antibiotic resistant activity is because of the presence of this extra chromosomal DNA. Whereas these 2 micron plasmid, we really do not know the function, and because of the presence of these plasmid, this will not have any additional character or a beneficial character to this particular yeast cell. But still, These 2-micron plasmid is very important in understanding the recombinant DNA technology, especially while creating various vectors in a yeast cell. Apart from the huge genomic size, so there are 312 genes have been completely mapped in this 16 chromosomal segment. Okay, now between the 16 chromosome and the 2-micron plasmid, so there are typical chromosome with nucleosome attached to that one, both in case of a 2-micron plasmid as well as this particular chromosome. Now the another major important function is going to be that the genes are not functionally grouped. What do you mean by this not functionally grouped? Probably you will understand very much with respect to the arrangement of gene, especially in a Forge vector. So you can always find that the head and tail genes are located on one side, then the regulatory gene for lysogeny located in one section, and the lytic cycle is located in another. So as a functional cluster, we used to find the genes arrangement in case of a prokaryotes. Whereas in case of higher organism in eukaryotes, so these genes are scattered across the genome. So you don't find, for example, the genes involved or the enzymes involved in a particular amino acid pathway. You don't find all of them grouped in one chromosome. So these genes are scattered across the genomic chromosome. So that is what is known as a functionally they are not grouped in fact. So this is the major difference between a prokaryotic cell, another one is an eukaryotic cell. We have to understand that there is a nuclear membrane, and here it is going to be a circular genome, but here it is linear chromosome. So there is 16 in number. 312 genes have been completely mapped with respect to the yeast. So this is number one. Now, before we get into the vector, we will try to understand the cell division in case of an yeast. So in case of an yeast, now the cell division is either by fission or by budding. So the very well discussed vectors are all in Saccharomyces cerevisiae, now which is the cell division is by the process of budding. So usually here also we have the cell cycle divided into G1 phase, synthetic phase, G2 phase, then the mitosis phase in fact. Assuming that this is the origin of the cell cycle, so now it is a well-defined cell with a nucleus and with a cell wall. So now this is entered into a synthetic phase. In the synthetic phase, basically what happens, the synthesis of nucleic acid takes place. That means the replication of nucleic acid takes place in the synthetic phase. At that particular time simultaneously, we also have a formation of a small bud. So now that is formation is takes place. Now then it is going to enter into the G2 phase. In the G2 phase, what happens? The synthetic phase is going to be almost complete. Now this budding started increasing. So during that particular time, the nucleus will also move very close to this particular membrane. So now that there will be a cell division. Now during this cell division, the DNA is going to be segregated between the two daughter cells. But in case of a budding, the distribution of cellular content is not same. So now most of the things are going to be present in the major cell. Now this is a budding cell, so it is going to be the distribution of the cytoplasmic content is not even. So but the distribution of genetic material is even, but this has to undergo a growth phase and it has to attain to this particular stage, whereas the other one will enter into this, so it will continue into this cycle, whereas the other daughter cell has to go through a developmental phase and come to this. Now this is the overall picture of how the cell division takes place in yeast. Again, I represent that it is made out of 16 metacentric chromosome. 312 genes have completely mapped. So now here I have just given this 2 micron plasmid. Now this 2 micron plasmid also contained nucleosome. Like that of a chromosome, you can find all histones and everything wound together in case of this 2 micron plasmid. But the interesting one is going to be that this particular plasmid, there are 50 to 100 copies per cell. Now, in case of an eukaryote, you have to understand that the replication, especially the genomic DNA replication, is only happens once per cycle. So it cannot have a continuous, for example, transcription, it can happen depending upon the requirement. Any number of transcription can happen. But when it comes to the replication, it happens only one time. After that, the next cycle, then I think it has to degenerate most of the enzymes required for the replication, or the most of the factors required for the replication. Whereas this 2 micron plasmid, that replication is independent of the genomic replication. So you are going to find multiple copies of this micron, that is 50 to 100 copies per cell. So now that is the major difference. So in order to have our effective vector should be introduced into the system, now that vector should also multiply like that of a genomic DNA. If not, then we are going to have a problem. So we will discuss all those things while studying about the various vectors created in case of an yeast cell. The next major problem is going to be the identification of a yeast gene after clone. In case of prokaryotes, the clone is easily identified using antibiotic resistant gene. But those antibiotic resistant genes do not function in an yeast, basically because yeast is not a bacteria. So as a result, antibiotics do not kill the unincorporated yeast cell. So now usually then they used to do by hybridization. So now either they will create an enriched cDNA or an RNA as a probe. Then they used to do the hybridization just to see that whether whatever the clone we are going to characterize, whether they really do have the inserted DNA or not by the process of hybridization. Okay, but the important features came in with respect to the complementation. This complementation is functional expression of our gene. So now here I think we are going to commonly use a terminology what is known as an auxotrope. So you remember in your first semester, I think in the techniques, so you also have various animal models or various organisms in which one of the topic is going to be the auxotrope. How these auxotropes are going to be useful. So usually auxotropes are nothing but a mutated microorganism. Either it can be a bacteria or even in case of an yeast. So now the mutated one is normally do not survive, basically because a critical enzyme or a critical gene product is completely mutated or deleted. As a result, the microorganisms, either the yeast, do not grow. But we can make them to grow in the presence of the replacement of that particular mutated gene. So then what happens, if that mutated gene is replaced, then this particular microorganisms used to survive. So that particular phenomena is called as an auxotropy. Now here what happens is that the most important one is that, so in case of an auxotropes, it is usually a mutant which is happening either in the pathways of an amino acid, an important amino acid, or a pyrimidine, which is a nucleotide component. So in case of an amino acid, if the amino acid is quite an essential, so then what happens, in the absence of that amino acid, the microorganisms, either the yeast or the bacteria, do not grow, basically because a key enzyme is lacking. Then if you replace that key enzyme, so then what happens, the metabolic process is going to be complete, so an essential component is going to be synthesized, then the microorganism or the cell do survive. Alright. Now during this particular process, so we do have auxotrope in yeast, we also have similar auxotrope in bacteria. Alright. Now suppose if there is an auxotrope in bacteria, then usually what happens, we have to replace that with a bacterial gene. But now here what happens that 20% of the yeast genes can be expressed in E. coli mutant. So I will discuss that probably in the subsequent table. That means say 20% of the yeast gene component or complement in nature, so that even if there is a mutation in a particular pathway, and that instead of supplementing the prokaryotic gene, we can also supplement an yeast gene in fact. So 20% of the yeast genes can be expressed in E. coli mutant. I think you have to understand what exactly is an auxotrope. Auxotrope, this is a mutated microorganism that cannot survive basically because a key metabolite is absent. Now you can make this auxotrope to survive by replacing that mutated gene. So this is very, very important. Now that is the basis for the identification of clone, especially in case of eukaryotes, that is by the process of what is known as a complementation. So the next will give us the clonal selection by complementation. I told you that we will study about the pathways associated with the amino acid or the pyrimidine. Now these are the most important gene product in an auxotropes, very well characterized and going to be very useful in the clonal selection. Now I have given you, I told you that 20% of the genes can be expressed in E. coli. Now this is an E. coli mutant. That means it is an auxotrope where the key genes of an amino acid pathway or a pyrimidine is mutated. And simultaneously this is the yeast gene component. Alright. Now this, for example, histidine B, if it is mutated, this function can be restored by replacing with yeast gene. Alright. Now we will come to this particular table. So now histidine 3, now that involves the gene product imidazole glycerol phosphate dehydratase or IGP dehydratase. Now this particular enzyme is very important for the synthesis of histidine, especially in histidine pathway. If the histidine is not produced, then this mutant do not survive. Then in order to survive that, so you have to replace this mutated gene. Okay. Now the mutated gene in case of an yeast gene is HIS3. If it is functional, we represent all of them with capital letters, HIS3. Now if the same 3 is written in small letters, HIS3, that means it is a mutated gene. It is no more functional. Okay. So now this is histidine positive and this is histidine negative, which is a mutated. So if you do write this in a small letters, that means it is a mutated, and this again represents the yeast gene product. Now the similar one in case of an E. coli, it is not the histidine 3, but it is histidine B gene. Okay. Now here all the E. coli gene plus or minus is written as histidine B plus for positive, histidine B minus for the mutated one in fact. Whereas here it is the capital letter and then the small letter. Suppose if there is a mutation in histidine B, now it has to be replaced with a good histidine B gene. If not, this particular function can be complemented with yeast gene. So that's exactly what I mean, that 20% of the yeast gene can complement E. coli mutant. Now this is with respect to the histidine metabolism. We have plenty of auxotropes both in yeast as well as in E. coli. The other important clonal selection is going to be the TRP5. So this is the yeast gene, functional TRP5. This is all small letters. It is mutated. Now this gene is represented by the tryptophan synthase, whereas the E. coli version is TRPAB. Okay. The next one is going to be the leucine 2. Now this is the functional gene, this is the mutated. The enzyme which is important is beta isopropyl malate dehydrogenase, whereas the E. coli version is LeuB. Now the another one is orotidine 5-phosphate decarboxylase, also called as URA3 or URA3, which is small letter, it is the mutated one. But in case of the E. coli mutant, the equivalent enzyme is pyrimidine F, PYRF. Another one is going to be the arginosuccinate lyase, ARG4. So now this ARG4 represent the mutated one. The equivalent E. coli is going to be the ARGH. Again, I will stress that 20% of all yeast genes can complement E. coli mutations. So we cannot use antibiotic resistant gene for the clonal selections. So here, in case of an yeast or in a plant cell or an animal cell, the clonal identification is based on complementation. You have to replace that with the right gene, then only we can identify that. So for that, auxotropes and identification of auxotrope mutants are very, very important.